Okay, we're going to start the lightning talks in about a minute or two. Just for the presenters, so I will keep track of your time. You have 5 minutes for your presentation. As you get 1 minute left, I'm going to show you this sign. And at 0, I'm going to cut you off, regardless of what you're saying. Sorry, because we have so many lightning talks. All right, so please stick to the 5 minutes so we can get through all of them in a reasonable time so we have a poster session afterwards. Sounds good? Sign. All right, okay. With that, I think we are ready to go. I think the first one is this one right there. Huh? Yeah.
Hello, everyone. My name is Laura Akey, and I am very excited to share with you today the Finger Lakes PFAS project. So our project is targeted water sampling for per and polyfluoral alkyl substances in the Finger Lakes region. It was motivated by PFAS becoming an emerging concern both for the scientists and the general public. We thought that in order to really address the problem that we have here, the first step is going to be to quantify the extent of the problem that we have in our local region. So here we have our map that we began with. We created a map on ArcGIS that includes potential sources of PFAS, including airports, fire stations, wastewater treatment facilities, and landfills. And we use this map to inform our sampling of the region. Once we get our samples that we collect and process according to EPA Method 537, we create our interactive map that you see here. Each circle is a sample that we have collected on the map, and when you click on that sample, you get our data, which includes the date and time, the source that we collect our sample from, our measured PFAS, which includes 17 different species, and the sum of those 17 different species, and then other metadata such as pH, the time from last rainfall, and our latitude and longitude. We have created this map in the hopes that it will be a user-friendly way to engage with the curious public who want to know about this issue in their surface waters. And we have a website that you can reach using the QR code and an email that you can reach out to. We encourage people to reach out with comments, questions, and concerns, suggestions. Nobody knows their local water better than the people who live there. And I would love to have you come to my poster and talk to me more about this, and I will take some questions now if there is time. All right, thank you.
Okay, thank you. Let's go to the next one. You know your order.
Hello everyone, I'm Emma Bland, and today I'll be speaking to you briefly about my master's thesis research that was completed here at SUNY ESF. So residential and agricultural runoff contributes to harmful algal blooms in the Finger Lakes region through nitrogen and phosphorus loadings within the watershed. In order to know how this nutrient flow is entering into the lakes, we need to know where these nutrients are coming from. This is made possible through processes like microbial source tracking, or MST, which uses bacteria that we know are really good indicators of fecal contamination from different types of animals and gives us an idea of how pollution is entering a watershed and contributing to deteriorated water quality. MST markers for human, ruminant, so we're talking about livestock and deer, avian or birds, and canine sources, as well as forms of nitrogen, phosphorus, and total suspended solids, were measured with the aims of evaluating whether the presence and concentration of specific microbial source tracking markers in streams can be used to predict in-stream nutrient concentrations and thus indicate biological sources of pollution, and how these associations vary between growing season and sample year. We sampled 8 Cayuga Lake tributaries historically known for nutrient contaminations, and this was a relatively long study that spanned about 3 years. When samples were collected from each tributary, they were processed according to EPA standard procedure for each analyte. They were then taken to the respective facilities for further laboratory processing. All nutrients were taken to Upstate Freshwater Institution, and the MST samples were taken back to SUNY ESF. Upon arrival at ESF, our water samples were filtered. DNA was then extracted from those filters, and then the extracted DNA was run through quantitative polymerase chain reaction, or qPCR, that allowed us to target the sources for humans, ruminants, avian, and canines. And then we used the listed analysis to answer the following questions. So were nutrient concentrations higher when MST markers were present in the samples? Did the nutrient concentrations increase along with the MST marker concentrations? And are there differences in these patterns over time? Specifically, are there differences between growing season and non-growing season and differences over sample year? So getting into our results, in regard to the MST marker concentrations and the detection of those, all measured and predicted nutrient parameters had greater concentration when the ruminant marker was detected. This was also true of most parameters with the other MST markers, with some exceptions that I have listed up here on the slide. The ruminant marker had the greatest detection at quantifiable concentrations, and they were approximately 5, 9, and 30 times greater than the human, avian, and canine concentrations on average. And for example, if we take a look here at Burroughs Creek, if I could go back. Oh, it won't let me. Okay, but at Burroughs Creek, we see much larger concentrations of the ruminant marker, indicating that this is a major source of fecal contamination to this water body. More importantly, oh, here we go. So Burroughs Creek, we could see that greater average concentration of the ruminant marker. Now, more importantly, when we show the linear mixed effects models at this high level of ruminant contamination here in Burroughs Creek and with the other tribs, we see that there was actually the high level of nutrient contamination was contributing to that excess phosphorus that has been a problem in this area for years. We also found that there was a human marker association with all forms of nitrogen that varied greatly by site, which tells us that there's a lot more going on within these watersheds that we were unable to account for in the modeling. When we looked at the temporal variation, we found all forms of phosphorus remained associated with the ruminant marker, and some forms varied between growing season and non-growing season, possibly indicating higher use of inorganic fertilizers during the growing season as phosphorus concentrations were greater during this time. We also found that there was some variation between sample year that was likely a result of varying environmental conditions. So major takeaways from this are we found that that relationship with ruminant fecal contamination and tributary phosphorus concentrations suggests that the ruminant fecal contamination is a possible explanation for those high phosphorus concentrations in the studied sites. And we hope that the results of this study may inform land managers of focus area for water quality mitigation efforts. Thank you. And do we have time for a question?
Hello, my name is Ulfet, and I will talk about a nutrient that we rarely think about. Yet it still shapes the future of our food, water, and planet. This life-giving nutrient is phosphorus, and it's an essential fertilizer. It's in everywhere. It promotes, it's in everywhere. It fuels food production, and the problem with the phosphorus is 75% of it located in Morocco, and we are using it so fast. By 2100, over 60% of the phosphorus reserves will be depleted, creating a gap between supply and the demand. What makes it more alarming is how inefficiently we use it. Over 80% of phosphorus fertilizers are lost to runoff, and it creating algal blooms. Just look at Lake Erie. Phosphorus-driven eutrophication caused the water pollution. And these algal blooms kills fish, destroys ecosystem, and it pollutes our drinking water. Its estimated total cost is around $100 million per year. So this life-giving nutrient is becoming life-taker. Our purpose is turn this life-taker back into life and back into life-giving nutrient. To do this, we are using waste materials like spent coffee ground and sawdust. We modify them for phosphorus adsorption. Here, upcycling has a crucial role because it lowers the waste and decreases the cost. After adsorption, we are trying to use this phosphorus-enriched material as slow-release fertilizer again to protect the nature and close the phosphorus loop. So we are using waste materials such as spent coffee ground and sawdust because these materials are abundant and biodegradable materials, byproducts with highly porous structures, enables them to absorb more material. When we modify these materials with cerium nanostructures, we create more powerful adsorbents because of the cerium's surface chemistry. Phosphate anions here can directly interact with surface oxygen vacancies of ceria, or it can ligand exchange with hydroxy groups of the surface. As you can see from the adsorption capacities, our materials can adsorb up to 20 milligram phosphorus per gram, even better than some commercial materials like bioxide. Here, the thing is not just about the numbers, it's about the potential of material. It provides rapid adsorption. We achieve highest adsorption capacities in 1 hour. It is cost effective, and it has high adsorption capacities, makes these materials scalable adsorbents for real-time use. So ultimately, we are using waste materials to adsorb phosphorus and reuse it as slow-release fertilizer. Here, cerium alone can enhance plant growth by stimulating photosynthesis, which it adds more value to our material. Thank you.
Room for question? All right, next one's up. Thank you.
Hello, I'm Atkia Fariha, and I'm a PhD student at Clarkson University. Today, I'll present our work on the development of an integrated and field deployable treatment system for the harmful algal blooms. Now, the harmful algal blooms or HABs are caused by excess amounts of nutrients such as nitrogen and phosphorus, which can be, as you can see from the left figure, from agricultural, stormwater, and wastewater runoffs. And one of the main microorganisms that cause HABs is Microcystis aeruginosa, that not only disrupts the aquatic ecosystems, but also produces potent liver toxins that can cause harm to both human and animal health. Now, our goal was to develop an electrochemical system, as you can see on the right figure, that used a nickel and antimony doped tin oxide anode and a stainless steel cathode with an unconnected aluminum plate placed in between them. While the anode was generating oxidant species such as hydroxyl radicals and ozone that can destroy the cells and the toxins. The aluminum plate experiences a bipolar charge, negative on the anodic side and positive on the cathodic side, and therefore undergoes induced corrosion and releases aluminum ions to the solution, which forms aluminum hydroxide, which binds with phosphate and removes it from the water. Therefore, our single-step system integrates simultaneous electrooxidation and electrochemical coagulation and focuses on both biological and chemical aspects of the harmful algal blooms. We also tested out two configurations, sandwich configuration and T configuration. In the sandwich configuration, we place the aluminum plate in the middle of two parallel distinct pairs of anode and cathode, and in the T configuration we place the aluminum plate at the bottom of the electrodes. Now I'll show you how our system worked. In the lab tests, in using synthetic lake water dosed with microcystis aeruginosa and phosphate, we observed nearly 100% removal of chlorophyll A, microcystin LR, and phosphate within 2 minutes using the sandwich configuration. So the sandwich configuration clearly outperformed the T configuration, as you can see from the left figure of the phosphate removal kinetics, and the T configuration showed less effectiveness, slower kinetics, and the slower removal kinetics. So on the right figure, we showed COMSOL simulations, mainly to visualize and to optimize this effect. Also from the simulations, we observed that in case of the sandwich configuration, the electric field was more concentrated and uniform for the aluminum plate, thus enabling faster aluminum ion release and phosphate removal. So after our lab-based tests, we are now moving on to scaling up this reactor to boat-mounted reactor capable of treating 200 gallons per minute in the field. And for that, we also did fluid flow simulations, as you can see on the right bottom. We will be placing baffles on both sides of the parallelly placed electrodes, which will concentrate the fluid flow through the electrodes, and the fluid flow will be uniform for the rest of the reactor. Therefore, we hope to deploy our electrochemical system in freshwater that have recurring harmful algal bloom issue. And that's it. Thank you for listening.
Hi. Let's start with a good scenario. Imagine a good sunny afternoon, and then you're having a glass of wine, but what you probably don't see beyond the wine is the wastewater generated by this wine. For every single bottle of wine, we could generate as much as 1.2 to 12 liters of wastewater, not just gray water. We're not talking about gray water here. We're talking about wastewater, acidic, with high nutrient concentration, with high contaminants. This is posing problems, not just problems of treatment, because for generating wines, for every 1,000 tons of wine, grapes harvested, you need about 3 to 5 cubic meter. Here in the Finger Lake region of upstate New York here, there are about over 150 wineries covering as large as 11,000 acres of vineyards, where this is not just very important, this is very important for the regional economy. But the main problem here is for every wine being produced, as earlier mentioned, there is a large quantity of wastewater. So what did we do here? We set out to understand this wastewater. Understand wastewater, the existing wineries, the existing treatment system, how effective are they? Then we asked some findings actually. From what you could see here, oh, sorry, I'm not done. All right. So from what you could see right here on the screen, so we focused, our case study was on wineries along the Finger Lake, as I mentioned earlier. Most of these Finger Lake, about 150 of them, each of them having individual separate treatment system. So you don't expect the same, output from this system. It is, of course, important to see the effectiveness of this treatment system. So we had a sampling, like daily sampling from, especially during the peak point. The main problem that was discovered was not because the existing treatment system was not functional, but during the harvest period, during the crushing period, it's just about a few weeks, there is a large quantity of wastewater being generated within this short period of time. And from findings, we discovered the existing system does not provide room to accommodate this large treatment at once. Because for the contaminant, most of these wastewater, as I mentioned earlier, they are high with COD, BOD, and the total solids were all considered. And at the end of the treatment system, at the end of the treatment of the existing system, some of the nutrients were reduced considerably. The contaminants were reduced also considerably. But during the peak moment, there is a sludge washout in the system. That is when, the septic system of, I don't have a pen. Okay, there's one. The septic system, sorry, is this okay? Okay. The septic system designed here, where you have this sludge, when you have a large quantity of wastewater coming in, instead of just this, because the quantity coming in at a very short period of time is very high, there is a sludge washout. What you would recommend for a fit-for-purpose treatment design? A fit-for-purpose means a system where, not just designed for a small quantity, but something that could accommodate the large quantity of wastewater that comes in, especially during the harvest period. Most of these charts indicate the first one here, for the COD and BOD. Then you have the TN and TP over there, considered all those ones. There is not a very clear reduction. There is not a very clear reduction in this contaminant in wastewater. The existing systems are working, but yet there is a room for discovery. There is a room for improvement here, which is why this is actually very important. So each time you take your glass of wine, you could be assured that your wine is leaving a better water footprint. Thank you.
Good afternoon, everyone. I am Shahadat Hussain, a PhD candidate from the ESF Chemical Engineering Department. Today, I'm going to talk about the cellulose nanocrystal, or the CNC-based thermoresponsive adsorbent, and the filter media preparation for the aqueous preface remediation. So by now, we all know that the negative consequences of the preface compounds on the human health and as well as also on the or on the aquatic environment. So to deal with that, we have seen the wide range of novel preface remediation technology development over the last couple of years or even so. So when we talk about the preface remediation based on the adsorption processes, to develop the adsorbent material, we really need to think about the couple of points. First of all, how sustainable the sources are that we targeted for the adsorbent material synthesis. Secondly, we need to think about how high the adsorption capacity onto our adsorbent material and how faster that kinetics for the preface removal from the water. Since we are talking about the capture of the PFAS molecules on the adsorbent material, we need to think about the regeneration capability or the desorption of the PFAS from our adsorbent materials also. At the same time, we are talking about the PFAS in the water, and we have seen that EPA has already limited the PFAS concentration in the water at the PPT, or even for some cases, for the some species below the detection limit. So how our adsorbent material is dealing with this low concentration of the PFAS at the environmentally relevant conditions. Finally, since we are talking about the adsorbent or the novel material development, we need to think about the economic and the environmental sustainability. So by considering all those points, we developed our adsorbent material for the aqueous PFAS remediation by making a sandwich adsorbent materials, which is PNI-PAM functionalized CNC. So to separate the CNC, we selected the sustainable sources forest residue biomass, which generated during the forest harvesting operation, which is abundantly available at the low cost at the northeastern region of the US. So we targeted that renewable lignocellulosic materials to produce the CNC, and we functionalized that CNC surface with the thermoresponsive polymer PNI-PAM with the aim of tuning the hydrophilic and the hydrophobic interaction to the targeted PFAS molecules. And when we tested our adsorbent materials in the adsorption processes, we found out that it has a higher adsorption capacity with the faster kinetics. And we tested our adsorbent at the very low concentration, as low as 1 ppb. And in some extent, it also dealt the PFAS concentration at the PPT level. And to find out the economic and the environmental sustainability of our adsorbent material, we also integrated the TEA and LCA with the whole PFAS, whole adsorbent synthesis process. Now let's talk about some of the findings by using our adsorbent. So when we tested our adsorbent in the batch adsorption processes, we found that our adsorbent was able to remove about the 80% of the long chain PFAS, which is PFOA in this case, and about 65% of the short chain PFAS, PFBA, from the water at 40 degrees C. When we, after the adsorption process, when we lowered the temperature at the ambient condition at the 25 degrees C, we found that our adsorbent was able to dissolve the about 82% of the PFBA and about 73% of the PFOA by only changing the solution temperature. Later, we translated that into the continuous process development. That is, we immobilized our adsorbent into the surface of the filter paper, cellulose filter paper, and we found that 18 gram per meter square adsorbent loaded filter media was able to remove about 70% of the PFOA and about 50% of the PFBA at the 40 degrees C, same as the batch adsorption process. When we lowered the adsorption solution temperature, our adsorbent media or the filter paper was able to dissolve about 76% of the PFBA and about 63% of the PFO. So the main key takeaway from those graphs are that our adsorbent and filter media was able to effectively adsorb for both short chain and the long chain PFAS materials. And at the same time, when we lowered only the solution temperature, it was able to regenerate or dissolve the PFAS compounds as well. So thank you, and we are also presenting one poster over there, so you can come for the more discussion. Thank you. If you have any question.
So, have you ever thought that a glass of water that seems clean and fresh could be bacterial contaminated and make you sick? Right now, over 2 billion people on this planet face the same question every day and not sure about their water quality. And here, our focus is the E. coli, the most dangerous pathogen, and the increasing microbial threat into the water contamination. So, as reported by the EPA, if we look into the map, the most eastern part of the America is highly exposed to E. coli. And the worst part is Continuous consumption of this E. coli contaminated water causes annually almost 3.5 million deaths. And the numbers are higher in the resource limited areas. And the reason could be that the current available methods, like the gold standard and the commercial available methods, they are slow, expensive, and lab limited. Even the research in this field is still trapped into the complex protocols and limited to the labs. So at this point, people need a method that should be simple, affordable, and easy to test their water quality on a daily basis. So by keeping this research gap in mind, we have designed a 3D printed color changing patch that works as a color tag and give you the E. coli concentration by the color change. So I will split this talk into the 3 sections. In the first, the mechanism and the validation. Second, the 3D printing patch design. And lastly, the smartphone analysis. So how our system works. So we started with the simple chemistry that when para-aminophenol react with the nanoparticles, it gets oxidized due to the reversible redox reaction and change color. So we chose a substrate that has a para-aminophenol and the glucose with the beta-galactoside bond right here. This substrate hydrolyzes in the presence of the beta-gal enzyme that is present inside the E. coli cell during the cell lysing. So when E. coli is there, these both things react together and release para-aminophenol, which then react with our nanocatalyst and generate color from colorless to the brown. So the intensity of this color change based on the E. coli counts present in the water. So next, we validated our system first into the solution. And here you can see the UV-Vis spectrum graph and a very nice peak shift at 380 nanometer, and the intensity of this peak shift increase with the increasing concentration of this bacteria. Next, we make a standard calibration plot into the solution with the different dilution of this E. coli, and here you can see that the color change is intense at the high concentration and start decreasing with the dilution factor. And from this system into the solution, we got a very nice limit of detection that is 41 CFU per ml, which is way lower than standard recreational water limit. So moving forward to the second step, for a 3D printing patch design, we need a material that should be biocompatible, holds the reagent, and absorb the water. So we chose alginate and gelatin, and infuse all sensing reagents inside the hydrogel matrix to design a homogeneous printable ink. Once the optimization is done, here you can see a 3D printing setup of our design patch. Once printed and dried, you can see a very nice thin film. So design patches are shelf stable and mass productive, which means that you can print almost 12 patches in just less than 5 minutes. So how our system works? Imagine you are dipping this patch into a water. If it stays white, it means safe. If it changes to brownish, means contaminated. That is warning situation. And the best part is our smart color card and AI-powered mobile application can capture the image, correct the background, and give you the precise E. coli counts right on your phone. Here is a image to show you how this actually works. So we dip the patch into a water, optimized water volume, and the gel soak the water and change the color based on the E. coli present into the water. So in conclusion, we have successfully designed a system that is fast, it works under 15 minutes, it's simple, everybody can use it, and affordable, that everybody can buy it without breaking their banks. So, healthy water is not a privilege, it's a right for everybody. And our $1 patch can make sure that every single drop we drink is safe for everybody. So think before you drink. Thank you.
Hello, everyone. So the final PFAS National Primary Drinking Water Regulation by EPA underscores the effective and scalable PFAS treatment in water systems nationwide. So in our study, we synthesized a cost-effective PFAS sorbent So it's called modified clay, MC, using a two-step process from naturally abundant clay. So when compared with 4 commercial-based solvents, our MC showed the superior removal efficiency and also the faster sorption kinetics for a broad range of PFAS, including short-chain and long-chain PFAS, as well as GNX and precursors. Additionally, under the environmentally relevant conditions, our MC showed robust performance under different conditions, including variations in pH, ionic strength, and NOM, indicating it can be sustained for the sorption performance in natural water systems. However, the MC has very fine particle size, which poses the practical challenges for separation in water system application. So therefore, to overcome this limitation, we synthesized another 2 novel sorbents from MC. The first one is magnetic modified clay, MMC, which incorporates the magnetite for easy separation using the magnetic field. The second one is modified clay beads, which engineered with larger particle size for practical application in flow-through system. So in the pure water spec with a broad range of PFAS, short-chain and long-chain PFAS, GNX, and precursors, the MMC shows very high removal efficiency. And also, when tested against the powdered active carbon, our MMC showed superior or comparable sorption performance in the natural stormwater. It further demonstrates that MMC can be practically applied in the natural water systems. We also evaluate the regeneration and reuse of MMC and MC by both chemical and photodegradation methods. So after the regeneration by chemical, which is methanol for MC and UV photo reduction for MMC, the both sorbents restored their high sorption performance in multiple cycles. And also to better understand their properties and the sorption mechanisms, we conducted comprehensive physicochemical characterization. So the FTIR analysis confirmed the CF bond after the PFAS sorption, and the positive surface charge and the spectral features indicate that electrostatic and hydrophobic interactions are the dominant mechanisms for the sorption of PFAS. And for the practical application in the flow-through system, we test MCB with the granular active carbon in the flow-through systems. In the drinking water, specs with 6 regulated PFAS. So while MCB showed slightly lower breakthrough volume than the widely used commercial GAC, it shows much better sorption performance at a higher concentration of 1,000 ppt. It reflects the higher sorption capacity for MCB. Another advantage of MCB is its higher cost effectiveness in the terms of the lower raw materials as well as less energy for production. So it indicates the potential for practical application in the continuous flow-through systems. That's all. Thank you.
Good afternoon, everyone. I'm excited to share with you a rapid and transformative approach to detect one of the most stubborn environmental threats of our time, that is PFOS. It's per and polyfluoroalkyl substances. PFOS are synthetic chemicals that are used since 1940s, with the application ranging from firefighting foams to Teflon coatings. What makes them so useful? It's their extreme stability. But that extreme stability also makes them most dangerous. Their carbon and fluorine bonds are nearly unbreakable, which gives them the name of forever chemicals. Today, there are over 15,000 known PFOS compounds, and their presence in drinking water is increasingly alarming. The US EPA has kept the allowable PFOA and PFOS levels at just 4 parts per trillion. That's like finding a drop in 20 Olympic swimming pools. The current detection methods, they rely on HPLC MSMS, which are highly accurate. Yes, they are highly accurate, but also they are expensive, time-consuming, and they are inaccessible to many labs. Testing can cost upwards of $200 per sample, and it can take weeks to return the sample. That's simply not viable when PFOS are found everywhere, in rivers, in rain, and even human blood. So there is an opportunity to develop a portable and inexpensive method to detect PFOS, where our solution comes. Our solution is PFOS Track. We introduce PFOS Track as a single-step, low-cost, portable electrochemical sensor that cuts through all that complexity. It's a true pre-screening powerhouse tool that is ideal for environmental monitoring, drinking water utilities, and even rapid response during the contamination events. Our PFOS track sensor works from 0.5 ppt to 1.5 ppb PFOS detection. And with the PFOS detection and the remediation market that cost over $5 billion US, tools like PFOS track offers not just scientific innovation, but also economic and environmental value. To close, in the world grappling with the silent spread of forever chemicals, PFOS track offers a bold, deployable, and democratized solution. We are developing our application and prototype with the sensor, and we are collaborating with WATTS, Water Technology, to commercialize our sensor. Faster, cheaper, and simpler. It's time we take PFOS detection out of the lab and into the real world. Thank you.
All right. Good afternoon, everybody. My name is Charles Rourke. I'm a technical assistance provider with the Drinking Water Source Protection Program, or DWSP2. Little pivot from research, but I'm hoping to give you all a primer on this program if you're not familiar with it and how it connects to some of the topics that we've been discussing today. So DWSP2 is a locally led, state-supported program. It's a volunteer program that any community or municipality with a public water source can apply into to receive free technical assistance to protect their source drinking water. The goal of this program is to give communities and municipalities a drinking water source protection plan that outlines and provides a step-by-step guidance on implementation actions to protect their source water tailored to each specific community. As we know, in New York, we pull our drinking water sources from a wide variety of different sources, from small confined aquifers or creeks that serve maybe a handful, 100 water customers, all the way up to large sources like Lake Erie, Lake Ontario, and the St. Lawrence River system. And because those sources vary so much, the potential threats and contaminants facing our communities and drinking water systems also vary tremendously. Some communities may be most concerned about point source contamination, things like chemical bulk storage, fuel bulk storage, pollution discharge sites, etc. They might be concerned about non-regulated sites like golf courses and land application of fertilizers, herbicides, and other contaminants, all the way up to land scale scope concerns about agricultural runoff, stormwater runoff, loss of ecosystem services from invasive species in forested watersheds. Because of that variability, our program steps into any municipality that applies and gets accepted to work through this process that you can see behind me to develop a DWSP2 plan. So just to give you a quick snapshot of what a participating community goes through during this process, we begin by assigning them a technical assistance provider that coordinates a stakeholder group. Typically, those are folks from the municipality, like water operators, town board members, other folks that are directly involved with the treatment and processing of source water, but also lots of different folks from the community to provide varied perspectives on threats that might face their drinking water. So folks from the county, from soil water conservation districts, from academic institutions like Cornell, SUNY ESF, Clarkson, and others, to provide, again, a lot of varied perspective and ideas on how to best protect that individual source water. We'll interpret the data, so we'll go through a water system overview, understand how the water is treated, understand threats that are facing it, concerns from the water operator and members of the community, and we start to put this data together into this DWSP2 plan. That includes free maps for the municipality to better visualize where their water is coming from, whether that's groundwater models or a watershed map, so that they can actually see where points of contamination are located in their source water area. All these maps and data are provided to the community for free. We then start to identify strategies with that full stakeholder group. What best actions can we take to best protect your source water? That might be reviewing permits for some of those regulated contaminant sources, all the way up to large-scale landscape projects like stream bank stabilization, sedimentation control, monitoring for invasive species. Each one of those often crosswalks a lot of other goals communities have to monitor invasives, to keep costs down for treatment, and to build a more resilient, healthy ecosystem around their source water. We also provide guidance on funding sources, so communities don't have to have this plan and not understand where they can find money and support to implement it. We provide grant opportunities and funding structures available so they can actually get right onto implementing these source water protection strategies. And lastly, a community that opts into this program gets continued implementation support even after a DWSP2 plan is completed and state approved. So it's not just sitting on its shelf, it's a living document, and they get the support from their technical assistance provider to see it through so that they're able to implement these actions. And I encourage you folks, I don't have the QR code up here, if you want to look on our website, I think we're the only acronym DWSP2 if you Google search it, to see if your community has an active plan and see if your drinking water is protected under one of these DWSP2 plans. And in the case that it's not, certainly reach out to your community members to apply during our next application program. And I'll say we rely a lot on active research being done by folks like yourselves and some of the grad work we've seen presented to better inform our best management practices that we share with communities. So we have a better volume and quality of data so these communities can make informed decisions about their source water. So that's a pitch for the program. Come by the little poster on the side and I'll be around for questions. Thanks.
Good afternoon. My name is Ryan, and today I would like to share with you the current approach for water quality management and how we can improve our decision-making process. The Soil and Water Assessment Tool, or SWAT, is a process model used to evaluate how investments in sustainable agricultural practices can reduce nutrient export. However, SWAT requires a large amount of data, expertise, and money to build, and model performance is often sub-satisfactory for predicting nutrient export. Prior studies that predict process model output using machine learning suggest these models could serve as a replacement for SWAT. Hydrologic response units, or HRUs, are combinations of similar land use, soil, and slope, and are the smallest spatial unit for which nutrient export is estimated in the SWOT model. Based on HRU physical characteristics and climate data, components of the hydrologic and biogeochemical cycles that affect nutrient generation and transport are estimated for each HRU, which are stored as HRU features. Once the HRU features are calibrated to the current watershed conditions using observed water quality data, the SWOT model can be used to evaluate the nutrient reduction associated with implementing best management practices, or BMPs. So the BMP implementation in SWOT result in changes to the hydrologic and biogeochemical cycles and thus changes to the HRU features. For this analysis, I chose random forest regression, which is a machine learning model that is flexible and can handle various data types. To train the random forest model, I used HRU features and nutrient yield from a SWOT model created by the USGS for a tributary to Lake Erie. This model was built to estimate total phosphorus yield under current watershed conditions, as well as under a management scenario aimed at reducing nutrient export. The random forest model was used to predict SWOT estimated total phosphorus using HRU features under two conditions. The first condition was to evaluate across the entire watershed where all HRU data was used, and the second condition was to evaluate on just the HRUs that were impacted by the implementation of management of the implementation of the BMPs, which is around 10% of the watershed. The target variable in the random forest regression model was the HRU total phosphorus yield, and the predictive variables were the HRU features. Before building the random forest model, I first performed feature selection of the HRU features to enhance model performance and interpretability. The random forest model was trained using 80% of the HRU data, and the performance of the model was evaluated on the remaining 20% of the data. For the first condition, the random forest model predictions were in strong agreement with the observed values. For the second condition, the random forest model had a lower degree of agreement with the observed values, meaning that random forest had a difficult time making predictions for individual HRUs. However, random forest predicted only a 1% difference in nutrient reduction between the management and calibration scenario as compared to SWOT, the SWOT estimated reduction. In conclusion, this analysis demonstrates that random forest can learn the relationship between landscape features and nutrient export. It also showed that there is room for improvement. One reason the model could have performed poorly for the second condition is that there were less observations to train the model. The overarching goal of this analysis is to develop an alternative tool that can perform as well as SWOT to predict water quality and also have the capability to evaluate differences in management scenarios. So for three next steps, first, I would like to evaluate machine learning to predict the HRU features. Second, the implementation of BMPs in SWOT would affect the HRU features in a predictable way because SWOT is deterministic, meaning that the same input will result in the same output. Therefore, I would like to evaluate the relationship between implementation of BMPs in SWOT and which HRU features are affected and how they changed. And third, for this analysis, I used the average of the HRU features across multiple years of daily simulations in SWOT. The results demonstrate that the relationship between landscape features and nutrient export could be learned from a single value for each HRU rather than daily values. Therefore, unless the goal is to simulate event-based nutrient export, the daily time step may not be necessary for the model used for water quality planning. Thank you.
Well, thank you all for the presentations. So the lightning talks are intended to be teasers for you to be excited to visit the posters. And so you can do this now with a little bit more drinks. And then at 5 o'clock, we're going to have a reception with more food and drinks to entice more discussions around the posters. So with that, thank you very much. Thank you, presenters.